Hello, everyone. Welcome to the Winter Quarter CDM Research Colloquium series. For the research seminar today, it is my great pleasure to introduce Dr. Taylor Ryder, who's currently a postdoctoral researcher at the University of Colorado School of Medicine in Denver. Dr. Ryder received her AB with honors from Harvard University in human evolutionary biology and a PhD from the University of California at Davis in food science under the supervision of Drs. Titus Brown and Ben Monpetit. Dr. Ryder's primary research areas of interest is in bioinformatics, computational biology, genomics, and food. Currently, Dr. Ryder is developing assembly graph-based methods to analyze metagenomes, to decontaminate metagenome assembled genomes, identifying microbial correlates to disease, and summarizing the content of a metagenome by taxonomy. Dr. Ryder also instructs and improves data carpentry workshops like Davis R users group and meet and analyze data help sessions. Dr. Ryder is the recipient of several fellowships, grants, honors, and awards during her time at Harvard University and the University of California, Davis. In her limited free time, Dr. Ryder likes to spend time in the Pacific Northwest hiking, biking, reading, baking, and drinking tea. I'm super excited to have Dr. Ryder at our colloquium to share her research work. The title of her talk is Metagenome Assembly Graphs and When to Use Them, a case study in identifying strain-level differences in gut microbiomes. Welcome again. Thank you so much for your time to be here today, and please take it away.
Thank you so much for that kind introduction. So today I'm going to be talking about metagenome assembly graphs. It's a topic that's sort of near and dear to my heart. And as John and I were just talking about before the seminar started, I think there's a lot of space for improvement in metagenome analysis space, especially using assembly graphs, borrowing from other fields, both within and outside of bioinformatics. So I'm going to talk a little bit about how we use assembly graphs and some space for improvements within that field. So to get started, let's see. Microbial communities are really everywhere. They inhabit every surface on Earth, and they do a lot of different things to impact those habitats. So they're involved in biogeochemical cycling or in plant health and human health. So there's been a lot of inquiries recently across biological fields looking into what these microbial communities do and how we can manipulate them or use them to impact the health of our environment or health of humans. And throughout this sort of, I guess, revolution in understanding the extent of microbial communities, there's really been extensive sampling across the world in different types of communities, and we've been using metagenomic sequencing to do this. So this was sort of a revolution, if you will, that started around 2000 or maybe a bit before that, where we realized that if we could sample in DNA from different environments on Earth, we could see a lot more microbes than just from culturing alone. And so through those sorts of inquiries, we've really expanded the tree of life and the different microorganisms that we know that exist on Earth. So for example, this tree here, these blue bands show uncultured organisms. So any point on this tree that overlaps with the blue band is a new organism that we've identified purely based on metagenomic sequencing and that we couldn't have observed from culture-based methods alone previously. And so these observations have really expanded what we know about diversity and then function of these organisms as well. So today I'm going to be talking about this concept of metagenomic sequencing, how we analyze this sort of data, how we use graphs to analyze this data, using graphs at scale, and then applying all of these graph-based methods to identify some strain level differences in gut microbiomes. So what is metagenomic sequencing data? The general concept is that we take a community of microorganisms And essentially we scrape them off of wherever they're living. We extract their DNA, which is a process of essentially throwing it into a blender, chopping it up into tiny pieces, and then getting it out the other side where it's shredded. And we want to try and reconstruct this genome from this pile of reads that's come out the other side. Sequencing data kind of comes in different flavors. So we have short read sequencing data, where if you have a genome that is 5 megabase pairs long, you'll get little tiny fragments of 50 to 350 base pair segments. They can overlap with each other or not, but we use these overlaps to reconstruct this genome. And then this other flavor is long read sequencing data, which is much longer, between 500 base pairs and 2 megabase pairs. And in the context of metagenomic sequencing, long read sequencing is still being pioneered, but it does stand to really transform the field. With these much longer segments, we can do a much better job at reconstructing these genomes. But right now, the current standard is short read sequencing. But one thing that's kind of exciting is the methods with assembly graphs that I'm going to be talking about today. While we're using them mostly for short read sequencing data right now, for the most part, a lot of the algorithms will work directly with long read sequences as well. So you can sort of just drop those in. So it is really a future looking technology as well. So if we get these metagenomic sequencing, these short reads, there's different ways that we can analyze them. And sort of the first one that's especially common in things like gut microbiome analysis, where we have a lot of observations of the organisms that we'll be looking at, is reference-based metagenome analysis. And so what this looks like is we'll have a database of organisms. This can be a database anywhere in size from 2,000 organisms or all the way up to millions of organisms, with complexity challenges scaling with the size of the database. But essentially what reference-based analysis seeks to do is take specific pieces of DNA in a metagenome and reassociate them back with a genome that's in the database. And so by doing this, we can usually figure out the taxonomy of the organism, so what species it is, or other organisms that it's closely related to, and sometimes as well the functional content, so what genes are contained within those sequences. But reference-based metagenome analysis does have some problems. So oftentimes what happens is when you map these short sequencing reads back against these reference genomes, you lose a lot of reads because they're not contained in the organisms within the database. This problem is reduced as your database size grows, and especially after a community has been sampled over and over again. But because of the complexity of the organisms that we're looking at, we never truly have every read that's in your metagenome mapped to the reference that it belongs to. And in practice, the fraction of this unanalyzable section when you're using a reference-based method varies a lot by both the tool that you use, which usually corresponds to the database, as well as the environment that the sequence comes from. So in things like the gut, some tools can have pretty good recall of up to about 80%, where this unanalyzable fraction is in red. But in things like marine environments and thermal environments that are less well sampled and are much more diverse, a lot of times 50% of the sample is unidentifiable, and this can actually reach up to about 90% of the sample for things like soil that are super complex with a lot of organisms that we haven't observed before. So sort of to combat this approach that drops a lot of your data is something called de novo metagenome analysis. And what this is you take these sequencing reads that you have and you're coming out your pile, and you look for overlaps in those reads or other signals in the data to reconstruct the genomes that they came from through a process called assembly and binning. And this works pretty well. It's actually how we've been able to identify a lot of those previously unobserved organisms and to expand the tree of life. But similar to reference-based analysis approaches, it also has some problems. And these problems come about in two different stages. The first is in assembly itself. When you look for overlaps between these reads, it works really well if you have sufficient coverage in the reads for every region in your genome, and if those regions in the genome are not super diverse. When they become really diverse, there's a lot of branching points where it becomes difficult for an assembler to make a decision about which assembled region to spit back out at you. And so you get a lot of unassembled reads. or you get a lot of really short assembled contiguous sequences, so about 500 base pairs to 2000 base pairs. And that would be okay, except that that becomes a problem for the genome binners. And so what a binner does is it takes these assembled contigs and it looks for other signals in them, like the use of specific base pairs or the read coverage depth if you map your reads back against these contiguous sequences. And then it says, oh, okay, I'm getting all the same signals from this group of contiguous sequences. They probably belong from the same genome bin. Let me stick them together. But this only works well when you have a contiguous sequence that's over about 2,500 base pairs in length. And so you can get both unassembled reads and unbinned contigs, all of which are ignored in downstream analysis. And similar to reference-based analysis approaches, we also have this data loss problem, and it differs based on the ecosystem that the microbiome is from. So again, in a less complex community, depending on the tool that you use, you can recover up to 80% of your reads from something like assembly, which is quite good. But then in a more complex community, you sometimes only get about 20% of your data analyzed. And so again, we're leaving a lot of data on the table by ignoring these fractions. So to understand how we can use graphs to lose less data, we have to understand a little bit about different analysis approaches. So metagenome assembly is that step in de novo analysis that I was talking about. So we take our bacterial genomes that we're interested in, and we cut them up into these short sequencing reads. And then assembly looks for overlaps in these sequencing reads and spits back out these different contiguous sequences. There's different approaches that these assemblers can use to look for these overlaps in the sequencing reads. So there's an overlap layout consensus method that literally just looks for overlaps in the tail ends of reads. And we can see based on this sort of toy example here, if we have 3 different reads where there's one variant in the read, it'll spit back out the consensus sequence produced from all of these different overlaps. This is an algorithm that some assemblers use, but it's a less popular algorithm. One that's more popular is what's called a De Bruijn graph assembly. And so what this does is it'll take our sequencing reads and cut them down into what's called a k-mer. And a k-mer is a word of length k in DNA. And so here we're cutting our sequences down to k-mers of length 3. So we take TTC and we get a k-mer out of that, and then TCT is the next one, CTA, and so on and so forth. So we can break our reads down into k-mers, and we do this for each of our reads. Then what we do is we look for overlaps between those k-mers, and the overlaps are size k minus 1. So we have CGA overlaps with the GA here. And so these 2 nodes would be connected by an edge. And so what happens when you get a variant, where here we have ATT that can overlap with either the TTC or with a TTG, is we get this bubble in the graph. And sometimes these bubbles come back together and they make a cycle in the graph. And then sometimes they don't, but in this case they do. And so this graph here in the middle is sort of an intermediate structure in assembly. And then what the assembler does is it walks the path that usually one that has the most coverage or depth from the sequencing reads, and it spits back out the most likely sequence. But this intermediate structure, the De Bruijn graph, holds all of the variation that's in the sequencing reads, and it's not lossy at all. So the sort of beauty of these graphs is, unlike the assemblers, which are forced to make choices in regions of sequence complexity, we can represent all of the sequencing data, no matter how complex it is, using these graph-based structures. So the De Bruijn graph has, each node is a k-mer, and so k of length 3, and then each edge is a connection between those k-mers. The compact De Bruijn graph is a simplified structure of the De Bruijn graph, where each of the nodes that are only linearly connected are collapsed down into a single node. So where in the De Bruijn graph you can only have a node with with sequences of length k, in the compact De Bruijn graph you can have any number of sequences as long as they were only connected by a linear path. And so the the compact de Bruijn graph ends up being the data structure that most assemblers use underneath, and then that a lot of tools end up working on because it's very convenient to be able to get back these larger chunks of sequences. So this is an assembly graph in the wild. It's a very complex data structure, and this is one from a mouse gut metagenome. So where the trade-off between using an assembly, which spits out your linear contigs, and then an assembly graph, the graph contains everything, but it's very complex, where versus the contiguous sequences don't contain everything, but they're far simpler to work with. But while we've had about 10 to 12 years of success with just using the assembled contigs, I think now the information and data loss that we're getting from only using assemblies makes the trade-off so that using these assembly graphs is much more worth it at this point to continue making progress in the field of microbiology and unculturable organisms. And assembly graphs, even though they're complex, they end up having these really nice properties. And this figure did not come through as clearly as I wanted it to, but it's another assembly graph of a perchlorate reducing community. And if we look at this assembly graph a little more closely, each of the regions of this assembly graph are highlighted in a different color if they ended up in a different sequencing bin. So if from computational methods, we inferred that they belong to a specific genome, it's colored in a different color. So here we can see there's this whole region of the graph that appears in bright pink, and that becomes one genome bin. Similarly, there's this whole region that's in this sort of blue color, periwinkle, and it becomes its own genome bin as well. Then there's these disconnected components, which are actually fairly rare in metagenome assembly graphs, but they also would have become their own bin. And so we can see that from this sort of representation that these assembly graphs are organizing sequences based on the organism that they came from, which is a super handy property of these assembly graphs. But the other thing that we can see is there's some sort of unleveraged information in these graphs. So for example, in this contiguous sequence, we can see that there's these different branches that are all colored in this dark forest green color, but then there's a bunch of gray branches as well that were unbinned sequences. So if we went back to the assembly graph, we could group these gray sequences with this dark forest green sequences and deduce that they belong together in a genome, but without using the assembly graph, that information would have been lost. And so that was sort of like a global view of what these assembly graphs look like of an entire population. But if we zoom in, these properties are also really useful at a more like a gene level scale as well. So this is the same perchlorate reducing community, and we're focusing in on one horizontally transferred gene. So a horizontally transferred gene is a section of DNA that gets swapped between different organisms. Sometimes it's organisms of the same species, or sometimes it's more distantly related organisms. But why these things become difficult to analyze is because if you have the same sequence that's shared between different genomes, it's flanked by different genomic sequences. And so it ends up giving you these really complex graph structures that are hard for assemblers to suss out. But what we can do with these sorts of graph structures is identify our gene of interest, and then identify the genomes off of these branching paths, and then infer that in an environment of salinity 5%, this PCRA gene is surrounded by Acetamincola, Anasulformonas, and an Acrobacter genome, or versus at salinity of 1%, it's only an Acrobacter. So it gets swapped around more as salinity increases, which is a nice thing to be able to detect. And this paper that put this out basically identified that they couldn't get this PCRA gene from an assembly, but by going into the assembly graph, they could find these structures and identify where this horizontally transferred gene was landing. And this is zooming in on a different tool that sort of takes advantage of the same properties to find antibiotic resistant elements and then their context within the graph as well. So from these sort of use cases, one can sort of intuit that querying into assembly graphs for your sequence of interest, whether that would be a gene that you're particularly interested in or a species that you're particularly interested in, is something that is interesting in order to get back all of the sequencing content that surrounds that sequence of interest. Once you have that sequence of interest, you can do things with it, but this is really computationally expensive to do. Yes, there's a question, I think.
Sorry, just a quick question. So you're starting just with the genome sequences, right, to construct the graphs?
So in this case, we're starting with the raw metagenome, well, the trimmed metagenome reads, so quality controlled metagenome reads.
Okay, so when does the gene information comes in? So is that, at what point these gene information get introduced into this graph?
Yeah, so that's a great question. And I think that's sort of an open area of research. There are two ways that you could address that. One is by taking your sequence of interest and querying into the graph. So if you say, let's say you're interested in this PCRA gene, there's different algorithms where you can essentially blast against the graph, or you can look for like k-mer overlap, or there's even now sort of an HMM model where you can look for these annotations. So, but let's say that you blast it against the graph. You're using a gene of interest to identify it in the graph, and you're doing that by a query. So that's one way that you could identify and annotate the graph. Another way is to sort of de novo annotate the graph. And in practice, that sort of looks actually fairly similar to taking your query of interest and either blasting it or looking for k-mer overlap, but it's sort of a separate step that you would need to do directly on sort of each of the nodes in the graph.
So de novo annotating a graph, are there other tools to do that? Or I haven't heard that term before.
Yeah, there are sort of tools to do that, and I'll get into this a little bit, but I'll talk about it now too. So there's one tool called PathRacer, and it takes the assembly graph that sits underneath Metaspades, which is a very popular metagenome assembly tool. And it uses an HMM algorithm to race across the different paths in the graph and annotate the graph. The problem with that tool is that the assembly graph that it's working on, the one that sits underneath Metaspades, has been heavily pruned. So it no longer includes all of the variation that was in the original graph. And oftentimes you're losing a lot of information by using that graph. But it's a super promising tool. And I'm hopeful that sort of the groundwork that was laid in that tool can be expanded back to complete compactor Boring graphs. Another approach that I'll talk about a little bit later is using a tool that came out of my PhD lab, Titus's lab, called SpaceGraphCats. And that one looks for, it simplifies the graph without losing sequence variation, and then looks for overlaps in k-mers between your sequence of interest, whether that's a gene or a genome, and then the graph itself, and it can annotate the nodes in the graph that way.
Okay, thank you.
Very much an open area of research that I'm looking forward to future developments on.
Yes.
Okay, so right, so doing this sort of assembly graph queries, we can find these sequences of interest, but in general, it takes a lot of computational power, and doing it at scale was generally prohibitive. Until some new tool development happened, and now we can do these sorts of queries at scale. So I'm going to go over a tool that I worked on a lot during my PhD, and I'm continuing working on in my postdoctoral research, called SpaceGraphCats, that does these queries at scale. But then at the end, I'll sort of tie in some other tools that are making this this type of analysis potentially available in a much more flexible way, and other sorts of growth that the field might soon see. SpaceGraphCats is a tool that makes this sort of querying into assembly graphs to find your sequence of interest much more computationally friendly. So to develop this tool, it was a collaboration between some graph theorists, Blair Sullivan's group and Felix Rydell, taking advantage of different properties of assembly graphs to make these queries possible. So I'll go over exactly what this algorithm does. So dominating sets underlie the sort of magic sauce of SpaceGraphCats. So what they do is they reduce the complexity of the assembly graph and then carve it up into to pieces. So these algorithms take advantage of a property of compact de Bruijn graphs. They're bounded degrees, so you can only have a maximum of 4 in edges and 4 out edges. And so by taking advantage of this, we can apply some pretty great algorithms from graph theory that otherwise would be NP hard, and instead they're not NP hard, and we can use them. So this is a toy compact de Bruijn graph here. So each of the nodes contains linear paths of sequences, so groups of k-mers that were connected by a single edge. And then each of the branch points are where there was sequence variation. And so what a dominating set is a group of nodes in this compact de Bruijn graph that make it so that every other node is at most distance 1 from a dominator. So if we look around this graph, we only have to traverse 1 edge in order to get to a dominator. And there are ties in this graph. So for example, this node here could either be a distance 1 from this dominator or from this dominator. And those are just broken at random, although it is deterministic. But what this does is it makes it so that where before we had a bunch of nodes, we end up with a bunch of pieces instead, and these pieces are much bigger than the nodes themselves. And so in practice, this is an E. coli genome here on the left. And if we apply this dominating set reduction, we can see that there's far fewer nodes after we do this reduction. And this just makes the graph a lot smaller and a lot easier to search while still preserving the proximity between different nodes. So one sort of use case for this sort of data structure is it allows us to query into it with a sequence of interest. And what we've used it a lot for is to do genome queries. And so what a genome query will do is it will take the genome of an organism that you're interested in, whether this is a bin that you've inferred from your own genome, or sorry, own metagenome in a de novo way, or whether it's in a reference database. And then you can use this genome and query into your compact de Bruijn graph and get back the entire piece. And the way this works is that any k-mer that's in the genome, if it overlaps at all with any k-mers within a piece, you get back the entire piece. And so basically what this does is it decorates out all of the sequence variation that overlaps with your genome of interest. So in practice, this works quite well to return neighborhoods of nearby sequences. And so what I'm showing here is Jacquard similarity between a target and then the fraction of the target genome that ends up being recovered. So if there's a very low Jacquard similarity, we don't get back any of our target genome, which makes sense. But once we get to about 10 to the -1, we recover between 80 and 100% of our target sequence. So this is a 0.1 Jacquard similarity, which corresponds to about a 93% average nucleotide identity if you were able to align these sequences. So once you have a genome that's approximately the same species, you can end up recovering all of the variation from that genome by doing one of these genome queries. So what this looks like on a real metagenome using real sequences of interest. I'm showing here 4 different genome bins that we use to query with. There's 3 different species basically, where these pink and blue are the same species, but just closely related organisms. So here's in the background is an MDS plot of the similarity between a specific gene that I'm showing. In this case, it's DNA gyrase A, which is a gene that's basically in every single bacterial organism, and it's only present in 1 copy. So we can use the similarity between those genes to compare how closely related 2 organisms are. And then this is all of the sequence variation that's represented by that gene recovered by the spacecraft CATS queries. And then in the triangles are the genes that were already in the genome bins originally. And then these are what the actual graph structures look like that represent all of these dots and sequences. And if they're outlined in these thick colors, they were in the original genome bin that we queried with. And if they're not outlined, then they're new sequences recovered by the graph-based query. So in this case, like for example here, this is all new sequencing information that we recovered from the query. And we can see that it's pretty complicated in its different cycles. And this is probably why an assembler wasn't able to spit this information back out for us, because there's too many tangles in this graph. And then this one was particularly fun because the original DNA gyrase A was missing from the sequencing bin, but when we did the query, we were able to recover it directly from the graph, thereby adding to the completeness or the full complement of genes that we observed in this genome. And while this is a universal marker gene that's present in every genome, over here we show that you recover about 13% additional functional content. So in things like accessory genes that you wouldn't necessarily expect to be present in every genome in a species, and certainly not every genome of bacteria. But this additional functional content would change our interpretation of what these organisms in the microbiome are doing. So we care about recovering this stuff so we can have a better idea of the function of these organisms and what they're doing in their community. So now that we can do these graph queries, we wanted to sort of apply this at scale to a problem that was of biological interest. And the problem that we sort of came up with was looking at strains in bacterial communities or in these metagenomic communities. So there's a lot of different definitions floating around for strain, but in this case, we refer to strain as any sort of subspecies grouping of organisms. So a lot of times we use a 95% sequence identity as the cutoff for a species. If you compare two genomes between each other and they're more than 95% similar at all of the places they overlap, then that's a species. But within species, you have all these different delineations where things can group more closely together, often based on their function. And so we're referring to any of those groupings as strain for the rest of this presentation. And the reason that we were attracted to this problem is because there's sort of this known entity in metagenomic sequencing data, especially within the human microbiome, which we've been able to study a lot, that while we can sort of identify up to 80% of our organisms present based on just mapping, we are missing a lot of stuff. And a lot of the stuff that we end up missing is sort of this 20% of hidden taxa and strain level diversity that some reads might belong to a species that we have sequencing data for, but we don't have sequencing data for those reference data for those genes in particular. And so it's just going unobserved, but we know that strain level variation is there. We know that strains impact biology. So we wanted to sort of see if we could use this technique to suss out this stuff. And the system that we chose to work on was inflammatory bowel disease. The reason we chose to work on this is because there's been a lot of research done in the past and there's a lot of publicly available data for it. What Crohn's disease, inflammatory bowel disease is it's a sort of complex of diseases. On one side, you have Crohn's disease, which is inflammation from mouth to anus, and ulcerative colitis, which is just in the colon, the lower large intestine. And the things that cause IBD are kind of unknown still. It's some intersection of host immune response, host DNA, the gut microbiome, and the environment. But when we were sort of looking at this, we were focusing just on the gut microbiome. And when you look at the gut microbiome, there's not a consistent signature. So this plot, what this is showing is inconsistency across microbial studies. So we have studies across the top, and then genuses of organisms and whether they were found to be increased or decreased in inflammatory bowel disease by these different studies. And across the board, there's just a lot of heterogeneity that has made it really difficult to try and find any sort of microbial signature that's associated with these different types of inflammatory bowel disease. But there's been a lot of research on this system, and we're kind of coming to this idea that this lack of consistent microbial signature might actually be in part due to strain level differences. So what I'm showing here is a specific organism called Ruminococcus navis, where one strain is associated with inflammatory bowel disease across different studies, and then another strain is not associated with IBD. This was found, this sort of association, by doing isolate sequencing and then mapping metagenomes back to these strains that were identified by this, this isolate and culture sequencing. So in isolate sequencing, you take an organism and you plate it out and do culture with it, and you can be pretty confident that you only have that one organism there. You go and sequence its genome, and the analysis of that is a lot simpler because the sequencing data is far less complex. And then if you have this really good set of references between the different strains, by just mapping against these strains, you can do a pretty good job of figuring out which ones are actually present. So that's how that association was discovered. And subsequently, it was found that this strain is sort of a bad bug in IBD and causes inflammation. So the reason that these authors were able to find that association was because they did this strain level analysis, versus when they looked at a species level analysis, there was no difference. When they looked at a strain level analysis, they were able to find increased abundance of one specific strain. So are there more of these strain switches that are occurring in IBD, and can we use graphs to help us find them? We used a bunch of metagenomes to do this meta-analysis. There was a total of 605, and they were roughly evenly distributed between Crohn's disease, ulcerative colitis, and non-IBD. And there was a whole sort of own research talk on how we selected some of these genomes that we thought might be interesting to look at for strain variation. But we picked 50 of them that we thought could potentially have some strain variation or be associated with inflammatory bowel disease. And we chose to dig in to just these 50. And then we used the methods that I sort of went through to reassociate strain variation from these metagenomes to our genomes of interest. So we took our metagenomes and we broke them down into their quality controlled reads, built the compact De Bruijn graph, selected a dominating set, and then with each of our 50 genomes, we queried into these and then recovered all of the sequencing variation out the back end. And in this case, we recovered the sequencing reads and then worked with those. So we had sort of carved up pieces of the metagenome, and we knew each of these pile of reads out the back end were a specific species that we were interested in. But then to find the actual pieces of the genome that were more abundant in inflammatory bowel disease, what we did is we combined all of the reads for one given species across all of our metagenomes, and we built a species level pangenome graph. And so we used a bigger dominating set piece for this, or a radius. So when we traverse the graph, instead of a node only being one edge away from a dominating set piece, in practice, we allowed it to be up to 10 edges away. And this gave us much bigger pieces. And essentially each piece had about 1 to 2 genes on it, which was what we were looking for. Then we were able to take these reads and look at the k-mers in them and see how much they overlapped with the pieces, and basically get abundances for each of the pieces in this graph. And once we had abundance information, it was pretty easy to do a differential abundance estimation to compare which pieces of these genomes were more abundant in inflammatory bowel disease or not. So this is sort of in summary. We had our communities, we did the metagenome assembly graph, got the species level neighborhoods, and then combined it to a metapangenome graph. I don't like that name, but it's what we're sticking with for now. This is showing that the algorithms can have a larger radius. So instead of just traversing one edge, you can traverse multiple edges to get to your dominating set node. And that gives us a much larger piece, which gives us more variation back from our graph. So when we did this for all 50 of our species, we saw some fairly interesting patterns. So I've broken it up here. These are our 50 species of interest that we looked at. We have Crohn's disease on the left side and ulcerative colitis on the right. We didn't look at all of the sequences. We only looked at about 10% from each species neighborhood. And then we compared their differential abundance. So if they're in gray, that comparison was not significant. When they're in brown, they were less abundant in Crohn's disease. And then when they're in blue, they were more abundant. So basically what we saw is that for the most part, we have a profound loss of diversity in inflammatory bowel disease, which is something that has been found in a bunch of different studies. So we were kind of expecting that. But for 5 of our species in Crohn's disease, we saw a very interesting pattern where you have increased abundance in part of the metapangenome graph, decreased abundance in another part, and then no change in part of it as well. And this is sort of a signature of what we would call a strain switch. So while portion of the genome is increasing, another portion is decreasing. And while the core genome that would be shared by all members of the species is basically staying the same. And one of these that we identified was Ruminococcus navis, which was that bad bug that I pointed out previously, that other people had already identified as having this pattern in IBD, but through isolate sequencing. So we were able to find this pattern through our metagenome graph analysis alone. And we sort of saw that this sort of shared fraction where some things are going up and some things are going down. Some of them are actually encoding the same genes, but with different sequences. So it's kind of promising that some of these sequences are for things like ribosomes, where you would expect that to be present and no matter which strain is in your metagenome, but that some of the sequences are slightly different shows that we probably do have different organisms that are present in this community in Crohn's disease. And we wanted to sort of make this so that it would actually be of interest to future research. And so what we did is we compared our sequences that were more abundant in Crohn's disease back against all of the different isolate genomes that are currently available. There's about 400,000 total isolate genomes available. So we compared against those and we found what had which genomes had the maximum containment for each of those species that were more abundant in Crohn's disease. And we can sort of make a suggestion that maybe it might be a good idea for inflammatory bowel disease to explore these specific isolates to see what properties they might have for disease. And one thing that was interesting about this is that this strain is one that was identified by previous authors using the research that I had showed you previously to produce an inflammatory polysaccharide. So it really is a bad bug in IBD. And we're thinking that maybe some of these other bugs might be doing something similar, where they might not be causing IBD, but they might be exacerbating it. But that's much more research, wet lab research that other people will have to take on. And I will skip that and just kind of go to here. I wanted to sort of make this note at the end. I think graphs are a really promising analysis technique to make sure that we're capturing all of the data in our system. And I think this sort of tool space is really developing. There's a lot more space for more developments, but even in human and reference genomes, variation graphs are making a lot of strides forward. And I think some of the lessons that they've learned in variation graphs, for example, I think one of the earlier presenters in this series showed that including only 10% of the variants from a group of genomes will make it so that you have more accurate mapping. And so I'm hoping that some of the lessons from variation graphs can be transferred back to metagenomes as we start using different graph analysis tools. And then there's also just these sort of promising tools that I'm hoping will help push the field forward as well. I think Spacograph Cats is great. I enjoy working with it, but it's probably not the tool to usher in all assembly graph analysis ever. And it's just part of the tool space. But promisingly, there's a lot of tools in this space. So I'm hopeful that it will continue developing. And with that, I thank you for your time.